Hello everyone. So in this video, I will be discussing the tertiary and quaternary structure of protein. So before we start, let's go back and have another look at the 20 amino acids. Last lesson, when we looked at the secondary structure and also the primary structure, we have focused on the backbones of the amino acids. And you can see here, every amino acid would have a carboxyl group on one side and an amino group on the other side, which is the backbone, and they all have that in common. What we're going to look at today is the side chains, or the variable Z group, R group. I think it's actually called Z in chemistry and R in biology. I don't care. I normally call it the side chain. And before we can start talking about the tertiary structure, we need to be able to classify these side chains into four categories. So you have non-polar, polar, acidic, or basic. Now you need to be able to do this yourself, but I'm going to just go through the 20 amino acids very quickly because there's just a few things I want to highlight, but make sure that you practice doing this yourself anyway. All right, so alanine, remember that we only focus on the side chain here. So alanine obviously has a non-polar side chain. It doesn't actually do anything. And then the next one is arginine, and you can see arginine contains an NH2 at the end, and this is an amino group, and the amino group is going to make it slightly basic. Remember from the titration topic, amino is a weak base, so it can actually gain an extra hydrogen ion, so it's basic. And if it is basic, it actually will be positively charged in water because it can gain the extra hydrogen from the surrounding. Okay, asparagine. Now there is a common misconception, and that is asparagine is basic, but it actually doesn't have an amino group because the NH2 in this case is part of a larger amide group. Okay, if it is part of a larger amide group, then it is really only just polar and not basic. So that's that. Moving on to the next group, which is aspartic acid. I think this is probably the easiest one to classify. Obviously, it has a carboxyl group. It's called an acid, so it's acidic. And if it is acidic, it is also going to be negatively charged in water because the hydrogen ion can be donated away by the acid. So it's negatively charged. Cysteine. Now, cysteine is slightly polar, slightly. I have seen quite conflicting reports on this. Some publication actually say it's non-polar, but it can actually form dipole-dipole interactions as well. So we're still gonna count it as polar. Glutamic acid, similar to aspartic acid, is acidic, and it is negatively charged. All right. So glutamine is the next one. Glutamine is C double bond O NH2 as well. So once again, this is an amine group and not amine, which means that it is polar and not basic. All right, glycine. Now glycine actually only has a hydrogen as the side chain. So we can just say that. So if the hydrogen is the side chain, then you can say that it is non-polar. The next group is histidine. Now if you look at histidine, it has this gigantic N and then NH, and it's really this N that can gain an extra hydrogen. So histidine is considered basic, and it can be positively charged in water. Okay, isoleucine, quite obviously non-polar. Okay. Similarly, leucine. Now, leucine and isoleucine are actually isomers of each other. As you can see, the only difference between them is the orientation of the carbon chain. So this is also non-polar. I mean, you can tell in the name, right? Leucine, isoleucine, isomers. Lysine is basic because it does have an amino group at the end, so it is basic, and it is going to be positively charged. Methionine. Now, methionine can be considered non-polar. It has a sulfur, but the sulfur is like buried between the carbon chain, so it can't really do much. So you consider that to be non-polar. Similarly, for phenylalanine, this ring structure is just CH in single double bond alternation, so it's just non-polar. for those who haven't seen this, this just means that you have C like so, and then you go like this. Okay. Proline. Proline actually looks really weird if you think about it compared to the other amino acid because it's not a linear chain. And it's important to recognize that in the case of proline, this part, this is the backbone. This is a C double bond, sorry, COOH and NH backbone, which means that whatever's left, so these 3 carbon actually makes up the side chain. And this side chain is nonpolar. It only contains carbon and hydrogen. Okay, the next group is serine. It's a hydroxyl group, so it is going to be polar. Serine as well, also polar. Okay. And then you have tryptophan. Now tryptophan, yes, it does have one NH here, but if you look at the entire side chain, it's the rest of the side chain is pretty nonpolar. You only have carbon and hydrogen for the rest of the side chain. So overall, this is nonpolar. And yeah, this N cannot get an extra H or anything. Thyroxine is polar. It does have a a hydroxyl group here. So this is polar. And valine is nonpolar. I don't know why that's solid and the rest is not. It's an artistic choice. It has nothing to do with anything. So nonpolar. So that will be the classification for the 20 amino acids. I think I show you here again, but this is obviously in the zeta ion format, and you can see the positive and the negative. And the charged amino acids. So it's charged because you can see how the amino group can gain an extra hydrogen, and then in the case of the acid, it can lose the hydrogen. All right, so if you have different types of side chains, you can have different side chain interactions. So the first one I want to talk about is the side chains between nonpolar groups, because I forgot to put the nonpolar here, but you have like 9 of the side chains being nonpolar. So the majority of the side chains would be in that group. And if you have 2 nonpolar side chains, then the interactions that they can form would be dispersion forces. And more significantly, let's talk about dispersion forces first. So if you just see one nonpolar side chain, and this is a exam question, for example, if I just give you a picture like this and I say identify the side chain interactions between these 2 amino acids, so you can see in the diagram that they both nonpolar, so the interaction between them will be dispersion forces. Okay. And specifically, you can, when you have nonpolar side chains, they will tend to cluster together to minimize contact with water, and this is called the hydrophobic effect. And we've talked about the hydrophobic effect before in the context of lipids. And essentially what it means is everything that's nonpolar is going to try to cluster together and away from all the polar groups. So for example, if you have a polypeptide chain and then these are water molecules, and if we assume that these are nonpolar R groups, the nonpolar R groups will cluster together, and that actually causes the chain to start to fold a little bit, and that is just simply so that they can be away from water. So just because dispersion forces are formed between them doesn't mean that it is not a very significant interaction. Especially if you have multiple nonpolar side chains, it can do a lot of I wouldn't say damage, but it can significantly impact the shape of the polypeptide chain. And essentially, if you have multiple hydrophobic effects and dispersion forces, that's going to cause the shape to be quite compact. And this shape is further stabilized by a range of other interactions in the side chains. So let's say, for example, if you have 2 side chains that are polar, so if we go with, I think everyone goes with serine because it's easy. Okay, maybe I pick something else. Let's go with serine. So you have CH and then CH3. So it does have an OH here. And then let's say this is part of a chain, and then here. I really should come up with let's be adventurous and actually draw something like, I'm gonna regret this. but I'm drawing thyroxine. Okay. And on this side, we have another OH. So you can essentially have hydrogen bonds between the side chains in this case. Or if another case is if you have cysteine being one of the amino acids, you know, I really should know what cysteine looks like. So this is S and this is H. And then if on the other side we, I don't want to draw, which one is the one that I haven't drawn? Let's go with one of the amines. So asparagine, for example, C, and then we have, try to remember this, C double bond O, and then you have N, H, H. So you could have dipole-dipole interaction between the sulfur of the cysteine and the hydrogen of another polar group, because obviously if it is polar, then we have negative dipole and positive dipole in these cases. Okay, so you can either have dipole-dipole interactions or hydrogen bonds if the two side chains are polar. So these are just examples for you there. The next type of side chain interactions that you can have is actually ion-ion, when you have ionic groups, which is basically mean that one of them is negatively charged and the other is positively charged. So I think I should go with like a really simple acid. So let's do aspartic acid, because I can't remember what the other acid looks like. Actually, it just had an extra NH2. Don't know why I say that. I can't remember. But you have C double bond O, and then O minus. So this is aspartic acid. And then let's say on the other side, and we're going to keep it with the same chain here. Oh, I can't remember any base. Is there any base that is not like 20,000 amino acid long? Probably not. So CH2, let's cheat and just put a, how many is this? 4. And then you have an NH3 plus. So as aspartic acid, and then this side we have lysine. So lysine is Lys. And you can see that you're going to have attractions between the negative charge of one side chain and the positive charge of another. So this is ion-ion or ionic bond, or it's an electrostatic force. It's not very as strong as like an actual ionic compound, because I mean the strength of the bond depends on the distance between the side chains. So it can vary quite significantly. So you have ionic interactions, or let's call it interaction. So I normally call it ion-ion. I think you can call it ionic bond. Just when you say it's a bond, it just sounds stronger, but honestly, there's no difference between the two words. And last but definitely not least is the unusual side chain between two cysteine. That's not how you draw cysteine. So two cysteine side chains. So this is one cysteine, and this is unique to cysteine alone. And then you have another cysteine here that has another SH. So basically, if you have these two side chains together, what tends to happen is the hydrogen is going to be lost, and they actually form a hydrogen bond. Sorry, not a hydrogen bond, a covalent bond between them. So you actually have this, and then another S, CH2, C. So you can form, believe it or not, a covalent bond. So this part here, this is covalent, and this is called the disulfide bridge, which is the strongest side chain interaction out of all of the one that we have just discussed, because it's actually a covalent bond. It's not just an attraction between something positive and something negative. All right, so disulfide bridge, and it's important to remember that cysteine is the only amino acid that can form disulfide bridge. Methionine also contains sulfur, but it cannot do this. So you can only have disulfide bridge between two cysteine molecules. So I did this drawing. Normally, if we're in class, we would do an activity. But let's say you have this is the primary structure of the protein. So the primary structure, you have this linear chain, and remember that the primary structure of protein contains the type, the order, and the number of amino acids. And when you have hydrogen bonds between the amide link, you have the secondary structure, which can give you, for example, an alpha helix. Now, if you think about if I have a few of these amino acids being nonpolar, then you can have something like this. So this is the clustering, for example, of nonpolar amino acids. So this is the hydrophobic effect. So you have hydrophobic effect here. Okay. And the acidic and the basic side chains may also be attracted to each other, so you can have some ion-ion interaction between the acidic and the basic side chain, and maybe the two polar side chain are also attracted to each other, so you potentially can have hydrogen bond here. So these multiple interactions will actually change the shape of the protein structure. So it's gonna allow it to further fold into a very specific shape. And what shape it's going to fold into depends a lot on the nature of the side chain, because if you don't have the first three amino acids having nonpolar side chains, then obviously you're not going to have this clustering anymore. So basically what I'm trying to say is the side chain interaction, it's going to promote further folding of the structure. So this is due to side chain interaction. So it's going to fold further from either alpha helix or beta, which is the secondary structure. It's going to fold even further, and it's going to fold into a shape that is unique to the amino acid side chain. So it's unique to the type of amino acids present. And this further folding, this structure is called the tertiary structure. So this is tertiary, this is secondary, and this is primary. Okay. So the tertiary structure, or the tertiary level of structure of a protein, is the folding of the chain into a specific three-dimensional shape, and it is caused by interactions of the side chain or the Z groups. And these interactions are still considered intramolecular because they still occur within one protein chain. So primary structure is just a amide link between the amino acids and the type, the order, and the number of amino acids. Secondary structure has to do with interactions between the amide links, and tertiary structure has to do with interactions between the side chains. And I'm sure the biology pupil know this already, but the side chain, due to these side chain interactions, the tertiary structure of a protein is highly specific, and it will depend on a specific amino acid sequence. So it's going to fold into a specific shape depending on what the type, order, and number of amino acids are. And this specific 3D shape is essential for the functioning of proteins, as we'll talk about in more details tomorrow. All right, this is another summary of actually the secondary and the tertiary structure, I guess, because you can see hydrogen bonds. So this is the beta sheet. So this is actually secondary. And then we can have alpha helix, which is also secondary. And then you have the different tertiary interactions. So you can have hydrophobic interactions, you can have ionic bond, you have polar, these are hydrogen bonds. These hydrogen bonds as well. And you obviously have the disulfide bond. Okay, so this is a quick recap of the four structures of proteins. So it's primary, which is just the chain. Secondary is when you start to fold the chain into either alpha helix or beta strand, depending on the hydrogen bonds between the links, the amide links. Tertiary structure is when that is folded even further, and these days there are so many programs that allows you to model the tertiary structure. And then you have the quaternary structure. Now the quaternary structure is basically when you have two or more tertiary proteins linked together. So quaternary is when you have more than one strand, so the interactions between the two strands can then be considered intermolecular again. All right, so this is just another definition of quaternary. So, and I give you an example quaternary structure here, which is hemoglobin, and it contains two alpha helix and two beta sheets. Now, this is just to show you, but quaternary structures, the subunits of quaternary structures do not need to be identical, and you can actually have many different tertiary structure proteins combined together, so it doesn't have to stop at 2 or 3. And I'm just showing some really beautiful quaternary structures of proteins when you have multiple tertiary structures linked together. And then that's that. I think that's it for this lesson. Thank you.